Now let's talk about the lab diagnosis of the E. coli infection. So whenever there we are suspecting infection by E. coli, we have to first always collect the sample or a specimen for diagnosis of any bacterial or for that matter any infection in the microbiology. So first we have to collect the specimen. So what could be our specimen? The specimen can be our stool. Stool whenever there is suspicion of the E. coli causing diarrhea. Then the specimen can be urine whenever there is suspicion of UTI being caused by E. coli. Then pus can be collected whenever we think that the wound infection has been caused by E. coli. Then pus can be collected. Then peritoneal was can be collected as a specimen whenever there is suspicion of the peritonitis by E. coli. Then sputum can be collected whenever there is ventilator associated edema being suspected to be caused by E. coli. Then we can collect CSF whenever we suspect that the meningitis is caused by E. coli. Then we can collect blood also whenever we think that the sepsis or septicemia is caused by the E. coli. So these are all the specimen in different types of infections where we are suspecting E. coli and collecting the respective specimen based on the type of infection. Now next we do the direct detection. So direct detection means we directly make a smear from that specimen and then we'll gram stain it or we'll directly see it under the microscope. Okay, so there we see after gram staining that there is gram negative bacilli with plenty of pus cell. Now here note here that we see plenty of pus cell in direct detection but not in the culture smear. Culture smear shows only the bacteria but not the pus cells. Okay, so please note this because pus cell is always present in the fresh specimen. Once the specimen is poured over the culture media, then these pus cells are destroyed, they are not seen anymore after that. Okay, so in the direct detection, we can see the gram-negative bacilli with plenty of pus cells. That will help uh, that will help us give us a clue about the E. coli or any other gram-negative bacteria, bacilli. Then culture. So the with the specimen we inoculate on the respective agar on some basal media because we know E. coli is a non-fastidious organism. So some basal medias are good enough to be inoculated with that specimen, of course. So then after inoculation we incubate at 37 degrees centigrade for 24 hours over the McConkey agar and blood agar. These are two basal media. Although blood agar is not a basal media, but still. In the McConkey agar, we can see pink colonies. Why? Because E. coli is a lactose fermenter. So we see pink colonies on the McConkey agar if it is caused by E. coli bacteria. I mean, if the infection is caused by E. coli, if we are suspecting E. coli, then it will be causing a change of pink color in the McConkey agar, and then in blood agar it will cause a change of non-hemolytic colonies, but sometimes it can also give the beta hemolytic colonies as well. Then after the culture colony characteristics are seen over the culture media, then with the discrete colonies we will make some culture smear and then we'll do the gram staining. So the smear is produced with discrete colonies on the culture media and then we do the gram staining and there we see the gram negative bacilli. Next we do the motility testing. So motility testing help us in differentiating between different bacteria as well. So for the motility testing we choose the hanging drop method. That is the most important method in microbiology for testing the motility. So there we see that the bacilli is motile because E. coli is a motile bacilli. E. coli is a motile bacilli. What are non-motile? Non-motile are Salmonella, Shigella, etc. Okay, those are non-motile in bacilli in the Enterobacteriaceae family. So in the Enterobacteriaceae family, uh, the Klebsiella, the Seginella, these are yes, Seginella. They are the non-motile bacilli in the Enterobacteriaceae family. But E. coli is a motile bacilli. So we will see the motility of the E. coli in the hanging drop method. Next, we do the identification. So identification can be done by conventional biochemical tests or by automated methods. Automated methods makes our life easy. By just putting the specimen, you will get to know about the organism. The machines are the Malditov and the Vitek, na, of course. So now come to the Biochemical test. So in the biochemical test you have to write the catalase test, oxidase test, citrate test, urease test, and indole test. So what are the test results? The test results are: catalase test is positive, the oxidase test is negative, the citrate test is negative, urease test is negative, and indole test is positive for E. coli. So I have written here P and 3P, but in exam you should not write this. Kokui is P and 3P. You should not write this. This is just to remember. You should write in detail like this: catalase test positive, oxidase test negative, citrate test negative, urease test negative, and indole test positive. You have to write in this way. In this way, okay? Not in this way. Not in this way. Okay. Then what is the IMVIC test? The IMVIC test is indole test, then methyl red, then VP test, and the citrate test. So indole test is positive. So let's write it here. Let's write it here. So I am VP and C. So it is P2N2. P2N2 means I is indole test is positive. Okay, that means indole test is positive and methyl red is also positive, then VP test is negative and citrate test is negative. So that is the meaning of the P2N2. You have to write in detail like this, okay? Not like this, not like this. So you have to, in the conventional biochemical test, you have to mention these all tests: IMPIC test and the catalase, oxidase, citrate, urease, indole tests. And then you have to write about the antimicrobial susceptibility testing, which is done for choosing the most appropriate antibiotic, which is to which the E. coli is most susceptible. And this is done on the by the disk diffusion method on the Muller Hinter agar. I have described this antimicrobial susceptibility testing in general microbiology. So that's all for the diagnosis of the E. coli. By writing these 6 to 7 points, you will fetch very good marks. Okay. And yeah, that's all for the lab diagnosis of the E. coli.